Hello, everyone, and welcome to this interview on IMI-Pain Care. My name is Melinda. I'm here from the European Pain Federation, EFIC, and IMI-Pain Care is one of EFIC's research collaborations. So we wanted to give the members of the project and the project leaders a chance to tell us a bit more about their project and to have an option for you, the attendees, give you the opportunity to ask any questions. You will see that there is a questions module at the very bottom of your screen, so please feel free to ask any questions at all. And at the very end of the presentations, we will be able to answer some questions. I will now hand over to Rolf Detlef-Trede. Thank you so much.
Good afternoon. My name is Rolf Detlef-Trede, and I'm working for Heidelberg University as a physiologist, and I'm the coordinator of the IMI-Pain Care project. What we would like to present to you in this webinar is who we are and what we do. The topic is a very broad one: to improve the care of patients for acute or chronic pain. And we do that with a defined set of topics that we are working on. So first of all, who we are. So it's a very large consortium. We have 40 partners, and they are the majority research centers and hospitals, but also pharmaceutical companies that are present in Europe. And very importantly, we have small entities, small companies that fulfill certain services to the European population. And particularly important, we have patient organizations and scientific societies also as members. And EFIC is actually one of the 3 member societies that we have. We are approaching the end of a 5-year period. And as you can see here, we cover quite a few European countries. We also have contributions from the US and Israel. And this has led to the possibility to also link up to activities, for example, in the United States that are then funded by NIH. So this is the who we are. Now, what we do can be summarized on one hand by these 6 objectives that we have. So one set is about postoperative pain outcomes and the activities that are nearing their end to define a core outcome set of domains. And there are also activities in the chronic pain field, in particular neuropathic pain. Another piece of activity that we do is to look at preclinical pain models and their translation to the clinic. This is true in particular for pelvic pain conditions, which have their specific problems, and therefore it's a huge gap to be filled. The IMI initiative of the European Union, together with FPR, is targeting better drug development processes. And therefore, we are also interested in enhancing the development process. And for that purpose, we look at translatable biomarkers that work in preclinical and clinical conditions, hopefully also clinical practice, that are responsive to pharmacological manipulations. However, we would hope that they could also be responsive to manipulations, for example, like psychotherapy and other things. We would like to project our insights to the European Medicines Agency, HTA agencies, etc. And one of the topics might be patient stratification, but we're also working on certain mathematical aspects of endpoint handling. And ultimately, and this is also an interest, of course, of the patient organization members, would like to support decision-making, clinical practice, provide algorithms, etc. And this event today is part of our efforts to disseminate our findings broadly. We would like to have your feedback and be able to discuss it. So you will have 3 more presentations that go into more detail, which are structured according to the 3 sub-projects that we have. One is on patient-reported outcome measures, particularly in the post-surgical field. Another one is on functional biomarkers, mostly electrophysiological ones, that show efficacy of medications in the different parts of the pain pathways. And the third one is going to be about improving translation in chronic pelvic pain. And I almost forgot to mention who will be presenting this. Winfried Meissner, who is the basically lead for the subproject PROMPT. Ombretta Caspani, the deputy lead for biopain, will be supported by Caterina Leone from Rome, who has led some of the efforts to identify the functional biomarkers. And Lysia Dimitriou will present some translation aspects on chronic pelvic pain. And then there's a little bit of a disclaimer that we will all show at the end and acknowledge the support that we have received. And with this, I think I would like to hand over to Winfried.
Yeah, hello and welcome from my side as well. My name is Winfried Meissner. I'm working as an anesthesiologist in Jena, Germany, Jena University Hospital. And as Rolf Detlef already mentioned, I'm one of the leads of the sub-project PROMPT. The specific characteristic of these type of project is that there are always two leads, one from what is called academics, that means from university, from research institutions, and one from industry. The other one is Hiltrud Liedgens from Grüntal. So we are always a team, not only on the top of the overall project, but also on the top of the sub-projects like PROMPT, and also on the top of each work package. As Rolf Detlef already has mentioned, I will briefly present the idea or philosophy of the sub-project PROMPT. I will not go into details. As you can see, this sub-project deals with patient-reported outcomes. Here you can see the included stakeholders or participants. We have academic partners, we have partners from pharmaceutical industry. We have, and this is something special, we have pain societies, not only as, let's say, institutions who somehow support this project, but they are real participants, so they are involved in the project. And at this stage, I would really like to thank EFIC. EFIC is not only presenting this type of seminar to you, but is really active with promoting the idea and the results of IMI-Pain Care on its meetings and with patient representatives, etc., etc. But also ESAIC and ESRA, two other large European scientific societies are involved in this project. And we have a couple of additional partners who collect the data. They are not all named here. Yeah, what is PROMPT about? I think everybody knows that the idea to measure pain currently mainly relies on patient-reported outcomes with all its problems. And I don't want to go into details, but you know that patient-reported outcomes on one side mirror the subjective nature of pain, or at least we hope that they will do. On the other side, there are quite a lot of issues concerning the validity, concerning the responsiveness of current patient-reported outcomes. And the overall idea or objective of our sub-project is to align or consent on a core set of outcomes, both in acute and chronic pain, which will not only work in clinical practice, but also in clinical trials, so that in the future there is, let's say, standard, or might be an accepted, worldwide accepted, standardized core set of outcomes when it comes to RCT in acute or chronic pain, and also when pain is assessed in clinical routine. This is the basic idea. there are 3 work packages, and I will a little bit go into detail concerning work package 2, because we already have some results in this work package, whereas work package 3 and 4 are still analyzing data and will achieve or will present their results in the coming weeks and months. So work package 2 is led by Esther Pogatzky-Zahn from Münster and also Hiltrud Liedgens from Grüntal as the work package leads, and it deals with prompts in acute post-surgical pain. Work package 3 with the same question concerning chronification of acute pain, and work package 4 with prompts in the area of chronic pain, specifically neuropathic pain and pelvic pain. Work package 2, again, the idea is to reach some type of consensus, which prompts might be used in acute positive pain studies as well as clinical routine. What is some of the elements of this work package are 2 large Delphi processes, which involve different stakeholders, including patients, patient representatives, to consent on first on the domains and in the second round on the measures of different patient-reported outcomes. And the other element, both element of work package 2 and 3, is a really large multi-center prospective study, which has aimed to assess a large number of different prompts, in other words, using different questionnaires. The aim was to include 4,000 patients in 4 different surgeries, so 1,000 patients for each of these 4 surgeries, altogether in 18 hospitals in 10 countries. What we achieved is to obtain data from 3,300 patients, which is, I think, still a nice result concerning COVID and all these issues in the last a couple of years. And there are some sub-projects in this sub-project. I only would like to mention one. This is in a subgroup of these prospective study. We also obtained objective mobility data by giving patients activity trackers. These fitness trackers. We used a more professional device, and the one secondary aim was to compare the objective mobility data with subjective patient-reported outcomes. There are also a couple of systematic literature researches on prompts for these different types of surgeries, and a couple of other subtasks. I will not show you all these subtasks, and will show you 2 examples of results. As I already mentioned, a large element of these work package 2 are 2 large Delphi processes. The first one was executed at the very beginning of the project and was on consenting of domains. And what happened here is a very systematic overview. So at the beginning, there was also literature research, and then a large discussion with different stakeholders. You can see it here on the left-hand lower part of the of this figure, and then the participants they decided on the domain. So here it's shown here. And this has already been done at the beginning of the project. And currently, the second Delphi process on the measurements is almost finished, and we will discuss the first results in the coming week with the group. This were the results of the first Delphi round, discussing the relevant domains. As I already said, 10 different stakeholder groups with 4 representatives for each of these 10 groups. So altogether about 40 people discussed a whole day and decided on those 6 domains you can see here, which were thought to be the most important ones in the area of patient-reported outcomes in acute post-surgical pain, both for clinical practice and for clinical routine, just as an example of these Delphi runs. Here are first results of the activity tracking device, and you can see here one, I would say, unexpected outcome was that we found almost no correlation between objective measurement of mobility of physical activity on one side and any of our patient-reported outcomes on the other side. The work package 3 is a little bit similar to work package 2, looking on potential prompts which might serve as indicator of increased risk for the development of chronic post-surgical pain. Again, this work package had executed some systematic literature review specifically for these 4 different surgeries we were looking on, and aims to develop a what we call decision support tool or predictor of chronic pain, which allows clinicians and caregivers to identify early patients with increased risk to develop CPSP. Again, we expect the results of this tool, of the analysis, which will form this tool in the next weeks. We already have some results, but it's a little bit too early to show them already today. We are discussing with this group the data. And the nice thing here is that we have such a large number of prompts obtained in this large prospective trial that we can really look also on some potential predictors which has not been looked so closely before. And finally, work package 4 is aiming to do a little bit the same in chronic pain, specifically in neuropathic pain and pelvic pain, and mainly worked with systemic literature researchers on one side and reanalyzed existing trials, a couple of existing trials, mainly from the pharmaceutical industry, which were given to this group and reanalyzed to identify prompts relevant for chronic pain, relevant for identifying interventions which really work in chronic pain. And again, here the group is in the late phase of analyzing the data so far. I cannot show you results, but it's only a couple of weeks, then results will be there and will be reported. Well, that's in a nutshell the idea of the sub-project prompt inside IMI-Pain Care. Thank you very much. So,
wonderful. Who would be up for the next presentation?
The next presentation, logically, is by the second sub-project that we have, which is biopain, which is represented by Ombretta Caspani and Caterina Leone, who are both sitting next to the same computer, and will present something on translatable biomarkers and some very interesting outcomes about excitability changes in people that can actually be measured. I don't know who of you will speak, but please start your presentation.
Okay, I will start first. I'm Ombretta Caspani. I work at the Heidelberg University and I'm the deputy of Professor Craig. And sorry, doesn't work. We are using Caterina computer, as you see. Okay, okay. there are two main regions that brought us to work together in the IMI-Pain project. And one is the inadequacy of drug therapy in the pain field. And in fact, in Europe, there are 64% of the patients that report inadequacy in the pain treatment. The other region is the low success rate in the drug development. In fact, and you can see here that in the nervous system, only 3% of the study that enter the phase 1 are successful. And this is the lowest successful rate in all area of the drug development. And this shows that the gap between animal and human in the drug development is even higher in the nervous system. So, and is also very high also in the pain field specifically. So, but nowadays there are different technologies, different methodologies that can be used in the nervous system to investigate the nervous system. They can be used in animal and humans, and they can identify biomarkers that can be used to evaluate the drug efficiency in clinical conditions. Here, you can see here on the bottom different methodologies that can investigate the different compartments of the nervous system. And the IMI-Pain aim to use the methodologies and even characterize the functional biomarker in the different compartments of the nervous system. We use assay that in our assay we use 3 different drugs that are known to activate the nervous system in the different compartment. And very important, we also use this biomarker in normal eye condition and also in clinical relevant hyperalgesia. And of course, we use it in preclinical and in humans. So here are, that is the powerful of having a big consortium like IMI-Pain. It allows us to design a protocol that was very similar in 4 different clinical trials. And each of these clinical trials focus on a different, use different methodologies and focus on different biomarkers. We have RCT1, and that focus on the peripheral using threshold tracking. And here the leader was the University of Aarhus with Nana Finnerup and Hatice Tankisi. And then we have the RCT2 that focus on the spinal and brain stem endpoint. And the leader was the Sapienza, the University Sapienza in Rome. And Caterina is the senior scientist together with Andrea Truini. Then we have the 2 clinical trials that focus on brain, the RCT3, focus on the EEG endpoints, and the leader is in the University of Louvain with André Moreau. And RCT4 that focus on fMRI with the leader that is at the University of Oxford with Irene Tracy. So similar methodologies were used from Lilly and the Bristol University later, and in the animal models. So here, what I wanted to show you is the design that has been used that was very similar in all 4 clinical trials. We have a healthy subject that came to our laboratories, once for the screening and again 4 times in each experimental days. The healthy subject receive either one of the drugs or the placebo. And here you can see the timeline of the experimental day. What is important to see is that we wanted to use normal condition, but also medical conditions. So we use HFX, high frequency stimulation, that induce hyperalgesia in one of the limb. And we took measures of the biomarker in the sensitized limb and the non-sensitized limb. Here, this pink bar represents the time for the recording of the biomarkers. And you can see that here is the intake of the drugs. So we did a recording before the drugs intake and 3 recording after the drug intakes. Then we also took 5 different blood samplings that allowed the PK/PD modeling for the quantification of the translability between species, but also for inferred dates for the compartments. And in addition to that, we also use prompts to investigate also the specific individuality of the sensitivity of the volunteers. So the data on our clinical trials are already collected, but we are elaborating that. However, together with the clinical trial, we also did a lot of side experiments. And here, Caterina can present us how all these side experiments were important to define the successful of the spinal biomarkers. So Caterina is on.
Okay, thank you, Ombretta. Hi, everyone. I'm Caterina Leone, and I work in Sapienza University, which is the sponsor of RCT2, whose leader is, whose PI is Professor Andrea Truini. And we performed several side experiments, and now I want just to show you some some results thanks to the side experiments we've done. Okay, so Ombretta said that we are introduced what was the aim of BioPain. We basically wanted to select biomarkers that could be used to develop new drugs. So they had to be translatable between men and animals. When it comes to the nociceptive system, the spinal cord, which was our topic, has been historically difficult to target for several reasons. So we have just a few strings to our bows, and we decided to focus on the R3, the nociceptive flexion reflex, which is a well-known neurophysiological tool, is a very old one, is considered the objective measure of experimental pain, and it's also considered the most reliable nociceptive reflex for assessing treatment efficacy. And importantly, it closely resembles the hin-paw flexion reflex, which can be recorded across species, so in any animal as well. And then we decided to use this somatosensory evoked potential measure, this cervical signal generated after stimulation, non-nociceptive stimulation of the ulnar nerve, which was a kind of a bet because it has been used a lot in neurological diagnosis, but for other purpose and not to study the nociceptive system and not to detect the spinal cord dorsal horn excitability. But since it closely resembles the intermediate potentials that can be recorded in animals, like the N1 that can be recorded in rats and other species, and is known in animals to be generated by wide dynamic range neurons that we know are involved in central sensitization, we decided to test if this signal could be a reliable biomarker of dorsal horn excitability in humans as well. What we did is enrolling basically healthy subjects and to induce, using human models of central sensitization, an interim stage between animal models and clinical trials. So we use these human models to which are we have many available human models, and we perform some studies on each of them, and the importance of having a clinically meaningful condition induced by the human models is indeed to create an interim stage between animal models and clinical trials. What we have to keep in mind is that all of them have different characteristics and different pharmacological profile. So going back to the R3, what I just wanted to briefly summarize what we knew before BioPain started. We just knew that thanks to the work by Garouche and Tertovara, we knew that the R3 threshold, the reflex threshold, was a reliable biomarker. There were uncertainty about the reflex size because the Danish group provide a lot of results, sometimes conflicting results. We knew that for sure low frequency ongoing afferent input to the dorsal horn was able to modulate the size of the reflex, but but there were uncertainty about a high frequency stimulation because it proved not to be able to modulate the size. And one possible open question was whether these human models exploiting a very painful nociceptive sensation could eventually be able to activate the endogenous pain systems that we know acts at the spinal level on WDR, wide dynamic range neurons. Therefore, it could have masked some effect on the facilitation. So we specifically tested during BioPain this hypothesis, performing an experiment using two different models, capsaicin and high-frequency stimulations. And what we concluded is that we were able to confirm the reliability of the reflex threshold as a biomarker of spinal cord dorsal horn excitability. And we confirmed that the size is very difficult to be modulated, but more importantly, we were able to clarify that the high frequency stimulation used to induce the conditioning sensation, is not able to activate the endogenous pain modulation, which was a question at the beginning. And these are the results on HFS. What we now know about our bet on the somatosensory evoked potential, the N13 component of the somatosensory evoked potential, we can say that our bet was paid back because it actually proved to be a reliable measure of dorsal horn excitability. We proved that during our side experiments that it can be modulated by the excitability induced in the dorsal horn using capsaicin. And importantly, that this modulation is prevented by the intake of a drug that specifically acts at that compartment, such as pregabalin. In a subsequent study, also within the BioPain consortium, we also proved that the N13 is modulated by the endogenous pain system, which we know is active at spinal level on the wide dynamic range neurons. So suggesting again that these neurons are a possible generators of this signal. So what we can, of course, it is very important to underline that, as I said before, the R3, the nociceptive flexion reflex, closely resembles the HINPO flexion reflex, which is well known to be a reliable measure at animal levels. But the N13 as well can be recorded in animals, and during our preclinical side experiments in BioPain, we were able to record, of course, not we, the other group, the preclinical group was able to record this response in rats. So what can we conclude? We can conclude that we were able to induce the clinically meaningful condition using human models that were able to induce an excitability at dorsal horn level, that this excitability at dorsal horn level could be detected using biomarkers that actually reflected that activation, and importantly, that those biomarkers are translatable between men and animals. So we can conclude that we successfully reached our goal. And thank you. That's it.
And of course, here we didn't list all the BioPain partners, but it was a huge collaboration between leader and PhD student, and there were a lot of names. And it was very nice and it is very nice to collaborate all together.
Yes, thank you very much. I left a note in the chat to all the participants that you're welcome to add comments or questions, either in the chat or in the questions and answer tab, and we will be able to answer them or discuss with you. However, before we do that, we have the third presentation. I saw Katie. Vincent has also joined us, who is one of the co-leads of the TRIPS subproject, but I think Lizia has prepared a presentation that she will now give. Over to you.
Thank you. Can you see my slides? Good. So this is a presentation on the third IMI project, which is the TRIP. And in this presentation, we'll focus on understanding the experience of chronic pelvic pain in women. I'm Lizzie, I'm the postdoc working on TRIP and specifically work package 9. So we have 3 packages in TRIP, and we're working on work package 9, and our main aim is to deeply phenotype women with pelvic pain to improve our understanding of endometriosis, associated pain, and bladder pain, and be able to give the relevant information to the other 2 work packages for the development of refined preclinical models for both endometriosis and bladder pain syndrome. And so work package 9 will address 4 main questions. First, whether we can demonstrate perturbations in the function of pain-relevant systems in women with pelvic pain compared to healthy pain-free women, whether we can establish biomarker profiles that are specific to endometriosis and bladder pain, and those that overlap both conditions, whether women with pelvic pain can be stratified into meaningful subgroups based on their clinical profiles and biomarker profiles, despite of their underlying diagnosis, and what are the key pathways that underline these subgroups, and finally, whether these subgroups relate to treatment responses. So in our studies, we have 5 different groups of people. So we have one group that's endometriosis only pain, and these are our inclusion criteria. So whether they have endometriosis, whether they have urinary symptoms, and what's their, whether they have pelvic pain. So our endometriosis only group has endometriosis, but no urinary symptoms, and has pain scores more than 4 on a numerical scale from 0 to 10. Then we have a comorbid group that has both endometriosis and bladder pain symptoms, and they tick all the boxes. Then we have a bladder pain only group that has no history of endometriosis, but they do have bladder pain. Then we have a pelvic pain only group that has no bladder pain symptoms and surgically confirmed no endometriosis, but they still score high on the pelvic pain scales. And finally, we have what we call a control group, but basically these are people without endometriosis or pelvic pain or bladder pain symptoms, but there could be other things going on with them. So this is the design of TRIP. We have the 5 groups that we selected from the NDocs database in Oxford and the BC database in Boston. And then a subgroup of the bladder pain participants were recruited at IBMC in Porto. And in phase 1, the participants had completed a set of questionnaires and given blood samples, saliva samples, and urine samples. And then in phase 2, they were asked to complete additional questionnaires and also a lot of the questions that they answered in baseline. The difference to this subgroup here is that these 2 phases happened at the same time because they were recruited separately. And then phase 3 was a subgroup of phase 2. It's when we had the physiological testing, and then in phase 4, which is an even smaller subgroup of phase 3, is the people who are going to have the MRI scan, and that's only going to be conducted at Oxford. And this is just the numbers that we have from each site, Oxford, Boston, and Porto, for each of the groups. So we have a good spread, especially from the Oxford and Boston sites. And these are the people that we have for phase 1. So these are the baseline questionnaire sample sizes. And we have, in total, we have more than 700 people. So now I'm just going to present a few of the results that we have. As I said, the baseline questionnaires, they're very long questionnaires, and there are a lot of results that you can find in our paper that's now as a preprint on medRxiv. But for today, we're just going to focus on their pain scores, specifically on dyspareunia, and have a look at how the pain affects people's quality of life. So we asked participants to give us a pain score for their different types of pain. So we have dysmenorrhea, pelvic pain that's non-cyclical, bladder pain, and pain during a gynecological examination. And these are the results. So on these graphs, we have the pain scale on the x-axis from 0 to 10, 10 being the worst imaginable pain. And then we have our subgroups here, and across the graphs, they're presented in the same order. So as you can see, that we have a spread of scores within each group, which shows that if you have an underlying diagnosis, it doesn't mean that you have the same symptoms as all the other people that have the same diagnosis with you. But more specifically, we see the endometriosis group scoring really high in dysmenorrhea and pelvic pain. The comorbid endometriosis and bladder pain group scores high across all 4 pain scales. And then the bladder pain group scores really high on pelvic pain, bladder pain, and pain during a gynecological exam. And then interestingly, we see the pelvic pain group only, so the people that don't have endometriosis or bladder pain syndromes, that they still give high scores, higher than 5, but they don't seem to suffer as severely as our other groups. So then we looked at dyspareunia specifically. So we had a section of the questionnaire asking about dyspareunia, that is pelvic pain during sexual intercourse. And we don't have the answers for the whole cohort, because some of the participants were either not sexually active or did not wish to answer to these questions due to the sensitive nature of them. But from the ones that did answer this question, we saw that more than 55% of the participants in each of the pain groups reported having experienced pain during or post-sexual intercourse at least once in their lives. And also about 50% of the people in each pain group reported having to interrupt or avoid intercourse altogether in the last 12 months. More than 30% in each pain group reported having dyspareunia either always or more than half of the times during when they had intercourse. And then when we looked at more closely into the location of this pain, we saw that there is a lot of variation, and most participants report pain either at the entrance or deep inside the vagina or in the abdomen or pelvis. So at this stage, we couldn't identify a pattern. And then when we asked them to score the pain on the same scale that we've seen in the previous slide, again, we see that the comorbid group is the one that scores the highest compared to the other two, but also the bladder pain group and the endometriosis group. They have a lot of people scoring really high on this one. And again, there is a variation within the groups. So we see a few people that don't experience this, but most of them score above 5. So moving on into more general quality of life. So we asked participants whether their pelvic pain impacts their daily activities or work. And so what we see is kind of a reflection of what the results we saw for physical pain. So we see that the comorbid group and the bladder pain group score higher on these pain impact scales. So they report having more negative impact on their daily lives and work. And we believe that it's higher than the endometriosis group also because of the nature of their symptoms. So having to worry a lot about going to the bathroom or having or being in a place where they can have a bathroom close. And then the pelvic pain group, even though we saw that they do have pain, it seems like they're impacted less than the other groups. And then we looked at the SF-36 quality of life scale. So this is a more generic quality of life scale. It's not specific to chronic pelvic pain. And it has many different subscales, but for now we're going to focus on the physical functioning and the emotional well-being scores. And in this one, I should say that the higher the score, the better your quality of life. So you can see that the controls here at the bottom have for physical functioning, they score almost perfect, so almost 100. But then we see the comorbid group having the lowest physical functioning score, followed by the bladder pain group. And then in the emotional well-being, we see that all the groups are now further away from 100. And as I said, our controls don't have pelvic pain, but they could have other medical issues. And also the emotional well-being is a bit more generic as a measure. But even so, we still see that the endometriosis and bladder pain groups score worse, have worse scores on this scale, and specifically the comorbid group. So just to summarize the key findings from phase 1, as expected, we saw that all the pelvic pain groups describe high pelvic pain, different types of it, and also poor quality of life. However, we see that the comorbid group is the most severely impacted one. And finally, we have highlighted that dyspareunia is both a common and important aspect of chronic pelvic pain, and we're recommending that it should be more researched and get more attention both by researchers and clinicians. Thank you.
Well, thank you all 4 of you for the nice presentations. Seems like our audience is a bit shy. In the chat, I don't see anything. It seems like the Q&A part is also empty. Melinda, do you have questions or comments from the ethics side? Else I would have some.
No questions from us at this point, but yes, this is your final chance to ask some questions. So any attendees who want to find out more, this is your opportunity.
Mm-hmm. So while you work up the courage to type something into the chat, I can have a few questions to the presenters. So for Winfried, in a way, the activity tracker is a biomarker. And has your sub-consortium already come up with an idea what it means if that biomarker does not correlate with anything? Does it mean it captures something that is useful and not captured yet, or does it mean it's useless? Any ideas?
Yes, thank you very much. First, it is a biomarker, but I wouldn't say it's a typical biomarker for pain. It's a biomarker, I think, for a couple of or influenced by a couple of conditions, specifically in the acute pain area, but also in the chronic pain area. There is interestingly a very intensive discussion right now what role activity might play for pain treatment, and everybody who works in the chronic pain field knows that the idea in chronic pain treatment today is not necessarily to reduce pain intensity, but to regain normal function. And it's more or less understandable that some of these patients, when they move more than they did before, they have sometimes even more pain. But is this now a good or bad outcome? This is, I think, a very interesting question. What we will do now, and what I think is very perhaps promising, is we will also check if activity early after surgery might be a predictor for late surgical complications, or even for late pain or chronification of post-surgical pain. So
and I assume the sign is if people have little activity, that then they would have a poor risk, right? Yeah. Okay, good. So possibly some innovation, and it's also interesting that this is nicely translatable to preclinical work that is done in this case in TRIP, but has also similar aspects. Maybe if I move to the next topic. So Katharina, you pointed out that this N13, the funny thing, spinal potential, has not really been used very much in clinical practice. Does this mean it's not going to be useful at all, or does it suggest that maybe people didn't ask the right questions? And if you think about patients, so which patients would make sense to be studied with this technique to look for their spinal processing?
Well, thanks for your question. Yes, I didn't underline this specific topic, but at the present, regarding your first question, it has been used in the past in the neurological diagnostic workup for other purposes to study the somatosensory systems, but mostly the large myelinated fibers and not the nociceptive system. It has been used in patients maybe just once with non-conclusive results. At present, we can only conclude that it will be definitely useful at group level for the purpose of studying the efficacy of drugs, because we can since the increase in the amplitude that we were able to detect is very, very small, it's difficult to say that it could be used at individual level, and I wouldn't, it's difficult to think on which patients it could be used, because most of the patients with chronic pain have widespread pain, so we wouldn't have any contralateral site to compare with. For now, we can conclude that it can be used at group level. In the future, we don't know. We will see.
I think that's a very important caveat. So it's not for individual diagnostic purposes at this point. However, scientifically, it could be interesting. You mentioned widespread pain, and you had also mentioned that this marker is changed when the descending controls are changed. So it could very well be that it is a marker that could show which patients actually have this alteration descending controls and have hence an increase in this amplitude. And also there are some treatments that for which there's a little bit of evidence that they could enhance the descending controls. This would be pharmacological or even spinal cord stimulation. So some interesting aspects.
Of course.
Yeah.
In the future, for future perspective.
I'm sorry to interrupt. We actually have some questions now.
You have, that's good.
And we have one for the pelvic pain project.
Which is perfectly fitting. Please mention that. Yeah, good.
It says for the pelvic pain project, the sample size was calculated to have around 200 participants. What was the justification for the sample size is the question. It seems that different numbers were included between 70 and over 200. Lizia, would you like to respond?
Yeah, so the participants were recruited from existing databases. So we used our inclusion criteria to go back to existing databases and identify people that fit our groups. Of course, the reason for having not the same number of people in each group is mainly due to COVID that has affected our recruitment process a lot. So we were hoping to have a lot more bladder pain participants as well, but unfortunately we had to pause and stop recruitment at different points.
Yes, I think that warrants its own webinar research in the times of COVID-19. There was a second question in this message about the pain score graph control group. It had pain concerning the quality in the pain score graph control group. It had pain concerning the quality of life. The pain described by the control group, was it related to what type of pain? The question was non-pelvic pain or different?
Yeah, probably non-pelvic. Yeah.
Yeah.
And then, sorry, while we have you, Lizia, there's actually another one. Comorbid conditions in pelvic pain projects. Could those predict the trajectories toward complications such as long-term effects or response to treatments?
Yeah, I think that's a bit more clinical question. I don't know, Katie, if you want to take this one.
Isn't that the idea? Katie?
Yeah,
Please.
I'm very happy to look at that. I mean, I think in clinical practice, we're always a bit guilty of focusing on our own silos. So gynecologists will think about the pelvis, neurologists may think about headaches or whatever their specialty is. So I think one of the important parts of TRIP is that we have captured that broader data, and that will allow us to look at whether we see a relationship between multiple comorbid conditions and different factors of pain processing, for example. So my hunch is that absolutely those comorbidities are going to be really important, but we haven't completely finished analysing that data yet.
Actually, there had been a question about the N13, whether it could be used in other clinical contexts, I guess, for looking at pain sensitization. And I think, Katharina, we discussed that maybe the context is not so much neurology, but chronic pain conditions. I think that's what our viewer, Gina Caetano, suggested in there. Do you wish to add anything? Or I just answered yes to her question in the chat.
But I think I have already answered that question.
I agree. Yeah, I think we were in the process of answering that question.
We have
well,
one
Melinda,
final question.
good.
One
Yeah,
final
please.
question, which is about biomarkers. So maybe that makes sense to actually close with that. Can the risk of bias of biomarker use, can that be calculated or not? And another question was, what is meant about false positives or false negatives? If you could please elaborate.
Yeah. So maybe I pick up what Katharina mentioned. At this point, the biomarkers that we're looking at do not really work on an individual level. So we see group differences. So I think we would find a lot of false positives and false negatives if we do the association with a clinical condition. However, as Winfried Meissner pointed out, the biomarker might capture something different, a different dimension that our usual endpoints do not cover. And then a poor sensitivity or specificity would actually indicate that we're capturing a different domain rather than the other things. So it's probably more interesting to see whether the biomarkers give additional information, which would count as false positive, technically, if you do that analysis, rather than trying to have a biomarker replace the patient-reported outcomes, because patient-reported outcomes and their measures and questionnaires have been well developed. So I always encourage people to use them if they want to learn about the pain of patients. And I see these biomarkers as opening additional domains. We're running out of time, but maybe people want to comment on that.
Yeah, yes, I would like to comment on. Thank you very much for this question. My main issue with biomarkers, I think they will open really new possibilities. But the main issue with biomarkers is so far I cannot imagine any biomarker which somehow mirrors the affective or emotional component of pain, or what we call suffering. And I see the risk if we focus too much on biomarkers that we forget this, let's say, part of patients' experience in acute or chronic pain. And so I, like you, agree, biomarkers will be an important addition, might be even nice for RCTs to detect analgesic responses. But when it's about the whole picture of experience of pain, we always will use some type of prompts.
Yeah. Roberta, Katharina, Katie, or Alicia, any additions?
Yes, well, in fact, that was also the reason why in our design of the clinical trial for biopain, we introduce a lot of prompts. And we will see later on when we do all the analysis, also the correlation between the prompts and the different biomarkers.
Mm-hmm.
Mm-hmm.
And I guess I would add, in endometriosis, the search for a non-invasive diagnostic biomarker has been a really important part of research, because we have to do surgery to diagnose the condition. And to date, it's really been relatively unsuccessful. The promise that I see from biomarkers is a method of allowing us to subgroup our patients on the basis of some of the underlying pain mechanisms. But I would never think that that would take away from us actually asking our patients themselves what their symptoms are and what the impact is.
Katie, that sounds like a very good closing remark. Melinda, do you want to add anything?
I would just like to say thank you all so much for joining, the attendees, the panelists. It was a pleasure finding out more about this project. Of course, you can always find even more information on our website, the europeanpainfederation.eu, or www.imi-paincare.eu. So go ahead and look up more information about all of this. Feel free to reach out to us if you have any questions. Thank you so much, and enjoy the rest of your day. Bye-bye, everyone.
Bye-bye.
Bye. Thank you.
Bye, everyone.